I'm a radiologist with CRL and primarily am based at Abbott Northwestern Hospital. This is another event in the revamped Twin Cities Maimonides Society. And I want to thank both Minneapolis and St. Paul Federation for sponsoring us. Tonight's speaker, Dr. Haase, is a professor of microbiology and immunology at the University of Minnesota. As everybody is well aware, we've experienced a pandemic like nothing other in my lifetime, certainly, but certainly not in any generation of recent memory. It's affected all of our lives in a profound way. And so this event tonight is really to understand, maybe at a basic level, the microbiology and immunology of this virus and how it elicits such a cascade of inflammatory responses that can be deadly. So I am going to give the floor up to Dr. Haase. I would ask that people keep themselves muted, and if there are questions, please put them in the chat box, which I will monitor, and periodically we'll open up the chat box for questions. So without any further delay, take it away.
Ash, if you want to get started.
I'm just trying to share my screen. There we go. Get that puppy up. So can everybody see the first slide? The new coronavirus associated with human respiratory disease in China?
Yes.
Great. So what I thought I would do to start out with tonight is provide some background and a review of this, as I was saying, is unprecedented in our times in history anyway, COVID-19 pandemic. Story starts at the end of 2019, or literally almost at the end, at December 30th. There were a cluster of 27 pneumonia-like cases reported of unknown etiology in Wuhan, associated with a human seafood wholesale market. There was some thought that that might have been the route of spread. It turns out that wasn't the case. It was just where people had a lot of contact. By January 7th, a novel coronavirus had been isolated. And 3 days later, the genome sequence of that coronavirus was released to the world, basically. So that's the coronavirus phylogenetic tree. There are 7 human coronaviruses. 4 of them cause sort of common colds. The 3 serious ones that cause severe disease, SARS-CoV-1 and MERS-CoV, and now SARS-CoV-2, belong to the beta coronaviruses. They're all basically have origin in bats. So SARS-CoV went from a bat through a civet cat, uses this angiotensin receptor, converting enzyme receptor. It causes SARS-CoV-2, can be mild or severe. The camel disease, MERS, again, started at some point. The camel was an intermediate host, started with a bat, causes MERS. And then we have SARS-CoV-2, which is most closely related in sequence to a horseshoe bat. In fact, extremely closely related, about a 96% sequence identity. And there's a thought that it may not even have gone through an intermediate animal host, but in fact simply passed through some kind of adaptation into the human species. by February 12th, so the article on the title there, New Coronavirus, was published online on the 3rd of February. By February 12th in 2020, the disease was named COVID-19 by the World Health Organization, and the virus was officially named SARS-CoV-2 by the International Nomenclature Committee. This is just the picture as of last night. There were over 41 million cases in the world, 128,000 deaths. The US has an unenviable position of being at the top there with 8.3 million cases and over 220,000 deaths. Down below are two pictures of what's going on in Minnesota. The red line is Minnesota. You can see on the left that we're continuing to have an exponential increase in cases. The dark gray line is Wisconsin, which has had just an explosive surge of cases in the past few days. The right panel shows you the case rates, and you can see how we actually got pretty good control in the early summer, bouncing around 1% new cases a day, and you can now see that we're just continuing to climb to new highs, basically. So the infection is very much with us. If you look at a map of COVID-19 in the United States, there are 8 million cases, almost 218,000 deaths. You can see from the sort of heat map, Minnesota is not the reddest state on a bit, but it's certainly got its fair share of cases, and we're with epicenters around the country, particularly now some surging cases throughout the Midwest and particularly in the Midwest. So we now know the virus is spread primarily by a respiratory route. There was a lot of discussion early on about surfaces and fomites and things like that. It's very clear now that it's passed by exposure to respiratory droplets. The 6-foot rule comes from the fact that if you're more than 6 feet away, there's a better chance that you'll have less exposure to an inoculum. You need to keep also in mind contact time, less than 15 minutes. And sometimes the particles will remain in the air. These are aerosols, will be suspended in aerosols for a considerable period of time. And there's just a comment about the contact. It certainly can be passed that way. So good case for personal hygiene, but much less important than the respiratory route. And it is found in the stool, the blood, semen, and ocular secretions, and none of those sources are really known in terms of transmission. It's not my favorite slide as an infectious disease person, but it does capture what a sneeze looks like. You can see the droplets, larger droplets in this case, but there'll be a mixed aerosol or an aerosol mixed in. And for those of you who are thinking about N95 masks that have a valve to expel exhaled air, just gives you an idea what a cough looks like in terms of what passes out while you're protected. Other people in your nearby vicinity are certainly not. I think the big surprise for most of us who hadn't anticipated this, because we had not appreciated the fact that people are actually maximally producing virus, exhaling virus at about a day before they actually have any symptoms. So that the asymptomatic folks account for 40 to 45% of the new SARS-CoV-2 infections. And it's Tony Fauci, this is from a talk he gave at the National Academy of Medicine on Monday, but I've been referring to it as a stealth virus because of this initially unappreciated transmission of the virus when people are not aware that they're infected, and those that they have contact with are not aware that they're being exposed. And modeling suggests that about 50% of the transmission occur from asymptomatic patients. This is just from I think it's from a metro in Moscow, but basically, so what we have when we talk in terms of mitigation now for a virus that's passed in this way, stealthily passed in this way by the respiratory route, we really have distance, we have time, and most importantly, we have mass. the masks we protect one another. So, by wearing a mask, you probably will have risk, certainly not negligible. But if both parties are wearing masks, there's much less likelihood that you will expose somebody to the virus or that they will expose you. So it's one of the key things in our transmission armamentarium. I found this paper, since it is the major, the World Series right now, going back to the last pandemic 100 plus years ago, the 1918 influenza epidemic, where they got the shot of US baseball players wearing masks while playing during the pandemic. A little bit about the virus. It is in the beta coronavirus genus. Coronaviruses are our champion RNA viruses in terms of size. I won't bore you go through the details, but it's simply the largest genome, 30,000 kilobases, encodes not only 4 structural proteins that we're going to be talking about, particularly the spike or S protein that's involved in binding the virus, allow it to enter the cell, but lots of really sophisticated programs that the virus can launch to impair or delay your innate and your immune defenses, which allows it to establish a foothold. And the receptor is the ACE2 as the major receptor. So the spike likes on, it's a trimer, it is 3 parts. And one of the parts can either be up or down. It's the up side that allows it to bind to the ACE2 receptor. And shown on the right is the spike protein and an antibody. You can see the antibody binding to the spike protein. And that's why the spike is such an attractive protein target, antigen for immunogen for our vaccines, simply because by blocking or by binding and blocking the spike protein's interaction with what's called the receptor binding domain, you can prevent the virus from entering the cell. This is just a simplified diagram. Both SARS-CoV-2 and SARS enter via the ACE2 receptor, and all those Y-shaped molecules are simply antibodies directed against the spike that prevent that entry. So in the current vaccines that we have in our armamentarium, I'm going to concentrate particularly on Moderna's vaccine and the Oxford AstraZeneca. They're involved in what are called phase 3 trials. Phase 1 and 2 are small safety trials, and then a little idea about some efficacy. Phase 3 trial is a full-blown trial with, in one of these trials, some of these trials, 30,000 folks or so, random control trials. And you're looking for both a safety and efficacy signal with usually some pre-set level of efficacy for licensure. And all of these trials involve data monitoring safety boards, so that if there's anything that they become aware of, the trials are stopped. The Oxford and the Moderna, I think, have been stopped for a patient, probably very likely from an unrelated incident. These trials now for the Moderna and Oxford vaccines, I think we can be reasonably optimistic that we may have a signal about safety and efficacy probably within the next few weeks. And then it's a question of distribution and a very well thought out scheme for how we're going to roll these vaccines out. In addition to the antibodies that are raised by vaccination or in the natural history, the course of infection, there are also monoclonal antibodies. This is one I noticed that they actually have the antibody binding on the wrong site. But anyway, monoclonal antibodies are an exciting development right now. Both Eli Lilly and Regeneron have products in trial. Trump got the Regeneron cocktail. They're beautiful because they can be expanded in large amounts. And they can be used both for prevention and for treatment. And the title here, I think this was a John Cohen article in Science, may curb the pandemic even before vaccines, although I'm hopeful, as I say, for the Moderna product and the Oxford product. So there's a fairly stereotypic clinical presentation with fever, cough, and fatigue, and substantial numbers of people, folks. Shortness of breath is usually a sign, and it's shown on the right, of already focal pneumonias that tend to be subpleural, described as ground glass opacities. Arnie Breyer and I were talking about the AI program that is pretty good at diagnosing COVID-19 pneumonia, and we're not quite sure how it works. But those ground glass opacities were seen in about 40% of the cases in China, and are certainly a sign of an initial focal pneumonia. Some of the interesting novel findings in the clinical picture for COVID-19 are the loss of smell and taste preceding the onset of respiratory symptoms. So I guess it's partly a good news story in that in the Chinese first cases are 81% were described as mild to moderate. In Minnesota and throughout the US, this is pretty much holding true. Some around 14–15% tend to be severe, requiring hospitalization and supplemental oxygen and so on. And then there's the 5% critical level. The case fatality rate in the Chinese series was originally 2.3%. It's varied wildly in Italy and in the United States and New York. It's been all over the map. But as we've gotten better at treating this disease, for example, in the Bethesda Hospital, where we've been treating our COVID-19 patients, our critical patients, the case fatality, or rather the critical patients are doing really quite well. And I would reckon that the overall case fatality rate is probably hovering around 1% altogether. So it's not just the lungs and the nose that get involved. We've become aware of a hypercoagulable state. It's not clear yet for the heart whether or not the virus actually infects the heart, the myofibroblasts and myocytes, or whether there's some kind of inflammatory reaction. But myocarditis is certainly something that we see. Kidney injury is really quite common, and there is an increasing appreciation of brain symptoms, everything from confusion. The strokes is actually probably related to the hypercoagulable state. There is an anatomical route from the nose through the cribriform plate to the ophthalmic bulbs up here, and it's possible that that may be one of the ways that the virus gets into the central nervous system. So we've increasingly had to appreciate the impact of this infection more broadly and systemically. I'm in the group that's at the increased risk, older adults and people of any age with underlying medical conditions. Probably about 40% of US adults are susceptible to severe COVID-19. Probably easily the most common underlying condition is obesity, followed by diabetes, COPD, heart disease, and chronic kidney disease. And we're also coming to appreciate what's called long COVID, basically. So people recover, most people recover quite nicely, but there are many people who are sick for several months after the disease. And it's not at all clear what the etiology or the pathogenesis of that long COVID-19 is. It's certainly under study. Therapeutics. There are two recommended treatments at the present time. One is remdesivir, an antiviral, and the other is dexamethasone. And I'll come back to that. We haven't really gone over the viral life cycle and the potential targets now and in the future. So after the virus breaks into the cell, it enters, the genome is encoded. It's translated into what's called a polyprotein. It makes a very large protein that's then cleaved to make the individual proteins of the virus. So outside of the spike protein for to prevent entry from initiating altogether, some of the other attractive targets, particularly, are the RNA-dependent RNA polymerase that's shown down here in the corner. So this is the enzyme. It's actually a complex enzyme that copies and makes new copies of the RNA genome. And remdesivir basically binds and prevents the that process of copying the RNA into viral genomes. But you can see other potential sites. For example, there's a protease that's required to clip the polyprotein into the individual genes. That could be a potential target. And we'll talk a little bit more later on about some of the other potential novel targets or drugs that might be repurposed for that part. So in the large Oxford trial, dexamethasone improved survival by cut deaths by roughly a third, actually a third in critically ill patients, which is very good news. The mechanism is presuming, this is not probably entirely accurate, although it's reasonably recent. If you divide the clinical course of COVID-19 pneumonia into pre-symptomatic, asymptomatic, when the virus is infecting cells in the upper respiratory tract, and then it gets down in the symptomatic phase into the lung to cause pneumonia. And then there's this late phase, and patients, they actually appear to be getting better, and then all of a sudden they go downhill really, really rapidly. This involved, it's been called cytokine release syndrome, cytokine storm. Pathogenesis is actually not really known, and most of it's being extrapolated from what goes on in the bloodstream. So the picture shown here is a sort of conventional picture of what's called acute respiratory distress syndrome. And it's very likely because of the cytokines that are involved in the lung and that inflammatory immunopathology that the steroids have a beneficial effect. I'll be coming back to that when we talk about pathogenesis as a guide or a rationale for the timing and type of treatment for COVID-19. And I talked a little bit when we were talking about prevention about monoclonal antibodies. I'm actually quite enthused about these. They were originally developed by these companies that were developing antibody cocktails, monoclonal antibody cocktails that have proved very effective in the treatment of Ebola. And I'm quite hopeful that they will have a quite large impact in the near future in treating patients. So I want to end up with pathogenesis. One of the questions, I think it was Arnie raised with me, is why are most COVID-19 infections not so severe? Why are they severe? And this is actually taken from an article from the North Carolina group, Ralph Barrick and company. And it's what I call the nose first, the lungs second model of COVID-19 pneumonia. What they did is they looked at the expression of the ACE2 receptor from the nose and the upper airways down through the bronchi and all the way down to the alveoli and the lower airways. And you can see the diminishing level of ACE2 expression, and that paralleled a diminishing infectivity. So most of the infection takes place, likely, at least from the results presented in this article, because of the higher expression and susceptibility of cells in the upper airways, and particularly the nose, where the virus gets a foothold, replicates to quite high titers. That's when they eventually become infection. That's when you have at least some of the symptoms, along with sort of systemic manifestations or reactions to infection. So what this model proposed is that in most cases, lung infection is not the rule, at least initially. It starts out as viral aspiration when some of these infected, some of the infection from the upper airways is aspirated and starts a focal lung infection, shown here where we often see them in the subpleural location. And there the virus will cause infection of two types of cells, and I'll describe those in a little more detail in a minute. The type 1 cells are these very thin cells that allow for gas change, and the type 2 pneumocytes that make surfactant and have just been described as the defenders of the alveolus. So the reason that the type 2 pneumocyte infection is so important, and the reason that it's called the defender of the alveolus is follows. So the type 1 cells are these incredibly thin cells that spread out. They occupy roughly 95% of the area of the alveolar of the alveoli, and they allow the exchange of carbon dioxide and oxygen. The type 2 cells make surfactant that spread out over the whole surface of the alveoli. They lower the surface tension and they allow the alveoli to remain patent rather than collapsing on exhalation. So that cell is obviously important for normal lung function. Shown down below in the little diagrams down here is simply a reminder for me to tell you the other really critical infection as a defender of the alveolus is the type 2 cell proliferates in response to lung injury. So it again begins producing surfactant, but most importantly, it regenerates and more type 1 cells to resurface basically the alveoli to restore their function. So clearly, anything that harms these type cells is really critical. This is a slide that lays out the choice of potential interventions, prevention through antivirals and immunomodulators through tissue repair, based on the idea that the initial infection involves obviously the virus establishing itself, and preventives would be the thing to prevent that. But in the early infection, the first 2 to 7 days, on average of about 5 days before symptom onset, you have antivirals as shown as being effective here. Then you have this 7-day period when we were talking about the inflammatory immunopathology, severe disease, and the acute respiratory distress syndrome. The viral load in red here is being shown from the blood, and it's shown as continuing. In terms of the research that we've been doing, it's not really the case here. The little dotted line shows that there's quite a rapid decline actually in the amount of coronavirus that we can detect in the lung. And the implications of that are as follow. If you're trying to prevent the loss of type 2 pneumocytes, and it's virus first, then clearly antivirals will be important. But almost equally important, if virus is basically down to a residual level out here in this severe phase in respiratory distress syndrome, then clearly the effectiveness of antivirals are going to be greatly diminished. And it's at this point, since the pathology is mainly related to inflammation, that dexamethasone and inhibitors of the processes that kill the type 2 pneumocytes at this point, which are turning out to be two kinds of death called apoptosis and pyroptosis, that are driven by certain cytokines like TNF-alpha, that these anti-inflammatory inhibitors could be effective at this stage. So I wanted to describe just a little bit of the research that's behind that. In this section, Arnie had asked me to talk about what's going on in COVID-19 research at the U of Minnesota. And so this was a project that came to us from Italy with Luca Sciaffinella and Nikki Klatt's lab, who came and joined from Miami earlier this year, in a collaboration with my lab and Tim Shacker and Jody Arison and Garrett. So what Luca brought with him was 4 cases of fatal pneumonia from Milan, Italy. And in one of those cases, we discovered a focal pneumonia, just like the focal pneumonia that the North Carolina group had described. So the red box here is around a brown looking part of consolidation in this pneumonia. This is relatively normal tissue. But when we pick up the coronavirus SARS-CoV-2 RNA, what you see from this area, you see all of these red cells that are little grape clusters, basically, that are making virus. And you probably may not be able to see it, but they are bursting. Basically, this virus is replicating in type 2 cells, and it is just killing them, and they end up bursting. And that's shown down here. That big red shard of dead epithelium is colored red because it has the traces or the residue, the replication of the virus that's taken place at that site. And as you might imagine, this causes severe destruction of the alveolar structure, the structure of the lung that's required for normal lung function. But again, importantly, it has taken away the type pneumocyte that's so important for that function and for the regeneration of that function. This is a special technique. I'm turning the color green, I'm turning the color of the virus green here. So all of these green clusters here actually represent what infection really looks like in the lung. You can see these little clusters of cells. So basically, these are islands of infection where one infected cell gets started. It passes it on to other susceptible cells in the neighborhood, and it travels until it's involved larger numbers of cells. This is just a big cluster of those same cells making virus. And then you can see these 3 cells here right next to one another, again, indicative of the kind of cell-to-cell transmission that's characteristic of infection in vivo reality in the lung. These are just big clumps of virus, just to give you an idea of the amount of virus that's being produced. And this is kind of a mask of what actually it looks like. So this is actually an airway, and you can see all of the green stuff that's fanning out from it, representing first of all of the virus coming down, in this case from the upper airway, killing and replicating and killing the lining cells of the bronchi and bronchioles in this case, fanning out and killing replicating and killing all of these type 2 pneumocells. And all that's left really are these shards of dead epithelium representing the huge amount of virus that's been produced and the extent of destruction in the lung. So just to conclude that part, we think that in this early focal pneumonia stage, that's the time to be really leaning on the virus with whatever antivirals we have in hand, remdesivir, monoclonals, before we get into this later inflammatory phase. Some of the other highlights of the past 6 months or so or longer, university was orchestrated the state's fastest on-site testing for COVID-19. This was a collaboration between the Lab Medicine and Pathology and my department and the Genomics Center. As of October, the U has done 400,000 PCR diagnostic tests. We've also launched an antibody COVID-19 test. Mark Jenkins, who's the director of our Center for Immunology, teamed up with Fang Li. Fang Li is in the College of Veterinary Medicine. He had published on the structure, purified and published on the structure of the SARS-CoV-2 spike protein. So Mark was able to team up with him and very rapidly develop a specific test for the detection of COVID-19 antibodies that's now moved on to the U's Advanced Research and Diagnostic Lab under the direction of Amy Carger. Talked a little bit about the various ways of preventing the spike from locking on to its receptor with antibodies. But there are other ways to do this as well. So there's a College of Science and Engineering team led by Ben Heckel and Jonathan Sachs, who've been working with Alon Hirschhorn and Dave Ferguson on finding some way, as they describe here, trying to cover the lock and key of that interaction with some kind of crazy glue and prevent it. Let me see if I can explain it a little more clearly. So what Alain Hirschhorn has developed is a target cell that when it interacts, when the virus infects it, that cell gives off light. So you can put these in these plates and add a library of small molecules and then ask, are there any hits in that screen for something that blocked replication so that you wouldn't generate light? And they've already identified three novel compounds that again would be one idea and an approach to developing novel therapies, new antivirals against this virus. David Bulmore has led the studies on the pre-exposure prophylaxis and preemptive pretreatment for coronavirus, and did the first randomized trial that showed hydroxychloroquine has no benefit over placebo in preventing COVID-19. There's a project in surgery, Chris Tignelli and Matt Puskarnich, who've been working on repurposing losartan, the angiotensin II receptor blocker. It's a fairly complicated scheme, but the bottom line is that it's possible that it might have some effect on this severe disease in decreasing the amount of hyperinflammation. And along similar lines, David Ingbar in the Department of Medicine and the lung group have a project where they're trying to use mesenchymal stem cells to prevent inflammation. The stem cells not only differentiate into various useful types of cells, but they're quite effective in suppressing immune activation. And then I haven't really talked much about the immune response. This was just a cartoon that shows the virus life cycle, and then the virus being presented to antigen presenting cells, and through the help of CD4 T cells, evoking a humoral antibody response and a cytotoxic T cell response. Well, in addition to the cytotoxic T cells that can destroy infected cells, there's things, not natural killer cells can also be used, are also useful as an innate response. And Jeff Miller, who for many years now has had a prominent role nationally in using natural killer cells in cancer therapy, is working with Josh Ryan on the left here to try to repurpose a natural killer cell product and candidate to see what effect it might have in increasing host defenses and in clearing virus infection. So that's a preamble. It's at that point I'd be happy to take some questions and have a discussion.
I guess I'll start the question, which is we've noticed a lot of people with COVID-19 will present with pulmonary emboli, and it seems like doing CT scans should be done as a part of a contrast enhanced study to look for pulmonary emboli. Is the fact that people are getting pulmonary emboli related to hypercoagulability, or there's actually damage to the endothelium that causes blood clots to form in the lower extremities and then go to the chest?
I think, Rob, I think it's both actually. There's a hypercoagulable state, but there's also clearly evidence of micropulmonary thrombi. It's been hypothesized that it's due to virus infection of the pulmonary endothelium. We're not seeing much evidence of that, so I'm not sure what the pathogenesis is, but there's certainly microthrombi that could contribute to some of the pulmonary problems. But I think for larger pulmonary emboli, I think that's part of the hypercoagulable state that's manifested in, deep vein thrombosis and stroke and all those other manifestations.
Ashley, you talked about the ACE2 receptor site on cells. Has there been any studies that compare individuals and whether they all have equal number of ACE2? If you look at pulmonary tissue or nasal tissue or tracheal tissue, are the number of these receptors from one individual to another and comparable tissue equal, or do people vary tremendously in the receptors that they have?
This is an extrapolation. It goes back to studies that were done some time ago when people were trying to develop air-liquid interface cultures of cells. And they noticed different individuals would have, different amounts of receptors. So the conjecture is, yes, it's very likely that part of the genetics and part of the susceptibility is related to individual variation in that receptor. It can't be quite that simple. There are also proteases that are involved in activating that receptor, and there are other genetic susceptibility factors that we're just learning about right now. But that's a good starting point, I think.
I think there's a couple of questions in the chat box. Dr. Thorne wanted to know how accessible the Regeneron cocktail that President Trump received. Is that going to be widely available soon, or what's the status of that for the general population?
Not. I was truly winging it. He's president, he got access. There are trials that are going right now. You could get access, although you wouldn't know if you were the placebo cohort. You could get access to it. That's sort of the bad news. The good news is that those trials seem to be that they're progressing really quite rapidly. And I'm hopeful that reasonably soon, we'll get a read on both the Regeneron and the Eli Lilly product. There would then be a huge ramp up that would be required where for the vaccine, we got online for eventually billions of doses worldwide, basically, with a clear-cut commitment and plan for accessibility. It would take really a considerable effort to ramp up so that they would be generally available. There's a question here. 11 different strains have been identified. That's probably even an underestimate. There's a recent paper within the last few weeks. I think they did sequence. I think there were like 87 strains. So yes, there are. I'm glad I'm seeing the rest of the question where you're going with it. The good news is that there is no indication as yet of two of the things that we were most fearful about. One is that the vaccine, that antibodies directed against the spike would be ineffective in neutralizing the virus. So that doesn't seem to be a threat. There is what's called a D614G mutation that is now the most widespread. It appears to have an advantage in terms of infectivity, but it does not cause more severe disease. So both of the concerns about strain variability, which you're going to get with, of course, with any RNA virus. It's inherent in the way they replicate. Although the coronaviruses have this complex genome that have some proofreading capability, and the extent of their variation is substantially less. So there's a question from Dina about, I've heard the flu vaccine may confer some benefit in reducing severity of COVID-19. Do any studies regarding this? I don't know of a study relating the flu vaccine directly to its direct impact, let's put it to COVID-19 infection. The reason we're pushing so hard for flu vaccine is we don't want overlapping epidemics, pandemics, basically. So we're urging everybody to get the flu vaccine, and we have tests so that we can distinguish whether you have COVID-19 or influenza, but I'm not aware of any direct interaction or impact on flu immunity. Although, I'll take that back just a little bit. It could be, when we get infected, the first defenses we have are called innate defenses. People call it trained immunity. It can be called trained immunity. So that the innate response you have to another virus, and it could be flu, it could be measles, it could be polio, something like that, may sort of get your immune system in a state where it may be more effective, at least it's to some extent, in controlling SARS-CoV-2 replication. There's a question on ACE inhibitor treatment for patients with heart disease, harmful or beneficial? And can you comment on convalescent serum? Michael, I'm not really familiar with the inhibitor treatment for heart disease. I can comment on the convalescent serum. So there's quite a bit of enthusiasm early on for probably one of the oldest ways that we have of containing infection, which is to take serum from somebody who's recovered and has antibodies against the virus and use it to prevent infection or moderate infection in someone else. And actually, the EU has collected, and Mayo has an ongoing program on post-convalescent serum for prevention and treatment. The reason I'm more enthused about monoclonal antibodies is it turns out that the antibodies that neutralize the virus in post-convalescent serum vary enormously in quantity. There are patients who recover who have no antibodies. There are patients who have, some. There are some champions that have just super neutralizing antibodies. And the idea was developing these tests, which Rob Tyler and others have done at the U, to get an idea, could we have a set of serum in which we had predetermined those that would be most likely effective. And we still have ongoing studies. The beauty of the monoclonals is that, first of all, is that you can produce them, they're uniform, they have high levels of neutralizing activity, so you don't have that variable to factor in. And the other thing about, takes me back to the vaccines, is the good news about the vaccines that are in phase 3 trials right now is they appear to be raising neutralizing antibody titers at least as great, or if not greater than, the best of our convalescent sera. And there's a question from Ralph. Are the Russian and Chinese vaccines, have they been studied here? Not to my knowledge. The Chinese vaccines have been extensively studied. Most of the data on them look really quite good. They are actually being used right now, and we will have a extensive clinical readout, I think, reasonably soon. The Russian vaccine, as you know, Putin winged it. He just started vaccinating people. We have very little information on the Russian data, so we really don't know how effective they're likely to be, and that's why there's a push for, broad collaborative studies and shared data across the nations.
I have a quick question. Once there's a vaccine, how long will it take for the virus to die out?
Good question. So we're going to roll it out, probably healthcare providers. First, elderly, those at most risk. That could happen as early if we get the signal we want from the phase 3 trials in early 2021. maybe the more general public by summer, something like that. So you imagine, the herd immunity is when you're out at, 60, 70% of people that have taken the vaccine. It's really hard to estimate how long that will take. It will certainly not be before mid to late 2021. It would be my best. In view of recent reports, some patients are having documented reinfection. Could you speculate on why immunity may not be enduring? Did these patients have compromised immune systems, or may this be a harbinger of not good things to come? Well, to my knowledge, I think there are only 2 well-documented cases where we have genetic evidence of 2 distinctive viruses. These patients are being studied very intensively. To my knowledge, we do not know why they did not clear the infection. It's probably one end of the spectrum of long COVID. With people who are continuing to have symptoms after you can't find the virus in their nasal secretions. It may be that in the lung and the other tissues there is continued virus replication and inflammation. That's all speculation right now. There was nothing known about, one of the things we're worried about with patients with known immune compromised systems, that was not clearly known in this case. So we don't know. The good news is doesn't seem to be, there are only a couple of cases right now that clearly documented, but it doesn't seem to be a large and emerging population.
What other questions do we have? It sounds like we've had a very thorough discussion. It's 3 minutes to 8. Last call for questions before we close the meeting. Thank you so much for presenting to this group. This was outstanding. We really appreciate your time. I've heard several talks since the pandemic began, but none as comprehensive of this. So I really appreciate you coming to speak before this group.
My pleasure.
Thanks, Ash. This is great.
Really was.
Thank you. On to the debate.
Yeah.
Thanks everybody
Thank
for joining us tonight. We appreciate it.
Thanks so much.
Dan, thanks for getting it going. Jeff, if you're on the line, thank you very much.
Thanks, everyone.
Have a great night.